Welcome back, everyone. Today, we're going to talk about cellular membrane notes. For you guys that don't remember, my name is Mr. Olson. Let's get going, okay? So, first thing, before we even take any notes about the cellular membrane, we're going to start doing a lab, okay? We're going to demo something right away. So, give me 1 sec. I'm going to pause the video. I'm going to change the screen angle for you guys. We're going to talk about this little demo here. And welcome back. Today's demo, guys, it's pretty simple, pretty easy, but it's going to help you guys explain what's kind of going on with this lab, okay, or with these notes. So first thing we got here, we got 2 pieces of a carrot, okay. In this beaker right here, this is 100% saturated salt solution. So it's water and just a whole bunch of salt, okay. And here, this is just normal fresh water. So what we're going to do, we're going to start off, we're going to take a mass of this carrot, we're going to take a mass of this carrot, we're going to put them in their solutions. We're going to go through notes, and then at the end, we're going to come back and see what actually happened, and then we're going to make some inferences about what happened throughout that piece, okay? So let's start with the fresh H2O carrot. If I lay it on the scale, this is science class, guys, so we're going to do everything in grams. This comes out to be 5 grams, okay? You're going to watch. I'm going to take this. I'm going to drop it in there, okay? I'll make the screen a little bigger so you guys can see maybe a little bit easier here. Perfect. Then I'm going to take this same carrot right here. I'm going to weigh it again. This comes out to be 8 grams. So we're going to start off with 8 grams. Okay. I'm going to take this carrot. I'm going to drop it in. Okay. Now, you probably can notice right away that that carrot's floating. This carrot's sinking. We will make some inferences about that as we move on. But let's get back to all the different things about cellular membranes. So at the end of it, we can make an accurate inference and kind of hopefully make sense of what's going on today. Okay? Give me 1 sec. What's going on here? Oh, there you go. Welcome back. Okay, about cell membranes. Let's get this down in your notebooks. The first thing is that all cells have a cell membrane. It's going to be the outer layer of the cell. Besides in plant cells, guys, which have a cell wall, that's the outside layer, but we'll get to that as we keep going, okay? But all cells do have a cell membrane. Now, they do have some functions. One of their functions, it controls what enters and exits the cell to maintain an internal balance called homeostasis. So if you guys remember the term homeostasis, hopefully you do from the 8 characteristics of life, it means that we're trying to maintain a stable internal environment. So for a cell, they have to regulate what comes in and out, because if they have too much of something coming in, they could explode. If they have too much of things going out, they could shrivel up and die. Or they could let bad things into your body or bad things into your cells, which could cause sickness or death or disease, okay? So your cell membrane, guys, does have that function. It has to maintain homeostasis, and it has to make sure that everything that's balanced comes in and out. Now, they also provide protection and support for the cell. Guys, I like to recommend, or not recommend, but to look at a cell membrane kind of like a balloon. They are movable, they are flexible, but they do create support, and it does protect what's ever inside, okay? So it can move, it can bend, it can get squished, but it does have that same form and always bounces back to it, okay? You think about us, guys. We have skin, and our skin's made out of cell membranes, okay? It's made out of cells. We can press on our skin, we can move our cell membranes, but it always bounces back to where it needs to be, okay? It's not a rigid structure. Now, talking more about cell membranes. The structure of a cell membrane is going to be very key to understand and be important to know. So the first thing you need to know is that it's a lipid bilayer. So it's 2 layers of phospholipids. Now, for you guys that don't know what this is, let's just break the word down. The prefix bi means 2, okay? So it's bilayer, so we know it's going to be 2-layered. And lipids is just another fancy word for saying fats, okay? The fat we're going to be talking about here, or the lipid we're going to be using, is a phospholipid. Now we're going to kind of draw and show what this looks like, but remember that our cell membrane is made of a lipid bilayer. So it is 2 layers made of phospholipids. Now there's a phosphate head, which is polar, which is water-loving. And there's a fatty acid tail, which is non-polar, which means it's water-fearing. So inside of that, there's also proteins embedded inside the membrane. So if we take a little look at the image over here, okay, where my pointer is, we have 2 layers. Hopefully you guys can see it. We have a layer of yellow here. We have a layer of yellow here. Those yellow heads are going to be your phosphate heads. These little squiggly tails here are going to be your fatty acid tails. In real situations, guys, this out here is where the water is going to be located, okay, on the outside of the cell. This kind of makes sense, okay? Your heads are attracted to the water, your tails repel the water, so it keeps them in one singular spot. Because you have one source of attraction, one source of repelling, kind of just keeps them in that one layered spot. And then these big pink things right here are going to be your proteins embedded inside the membrane. This is going to come more into play when we talk about passive and active transport later in the week. Here's just another good image, guys, for you guys to realize. This is the head. This is the tail. This is the phosphate head. These are the fatty acid tails. This is polar. This is non-polar. This likes water. This does not like water. Okay, guys, I know it's not the most easiest thing to understand. I'm not probably doing the best at describing it. It's just one of those things you have to memorize. Okay, so understand that there's a head, there's a tail. One likes water, one does not like water. It's a phospholipid bilayer. Now, this model is a fluid mosaic model of the cell membrane. So how do we break this down? What does that mean? Like, what does the term fluid mean? What does the term mosaic mean? We understand what models are, but how do we correlate those together? So I want to start with this term right here, mosaic. Now, if you were in class, I'd be asking, what is a mosaic, guys? And somebody would eventually raise their hand and be like, it's a piece of art where they like break glass and they put a bunch of pieces of glass together to make a picture. And I'd be like, yeah, okay. So realistically, what mosaic means, it's a bunch of different pieces put together to create one whole thing. So like a mosaic art is like where they have those little glass chunks and little pieces, and they build this huge masterpiece. Your cell membrane is kind of similar. It's made of a bunch of phosphates, a bunch of lipids, bunch of proteins, and all these things that kind of come together and they make this cell membrane, which some of you guys wouldn't consider to be art per se, but it is pretty magnificent. Now, fluid determines on how it moves, okay? So being able to move fluidly just means that it's not rigid. So something like marker is something that's rigid. It doesn't bend, okay? It's stiff, okay? This is a rigid structure. Something like a wire, though, is pretty fluid. It can move, it can bend, okay? Your cell membranes are fluid, and they're mosaic. Guys, if our cell membrane was not fluid, we would be stuck like this and be like, okay, and we wouldn't be able to move. That'd be an issue. So our model or our body is made out of these cell membranes, out of these cells that are fluid and they're mosaic. So they're made of a bunch of different things, but they have that fluidity property that allows them to properly move and actually be able to bend and adapt with what's going on. Okay, fluid mosaic model. Now, just a little reminder, little refresher. The heads are polar, guys, and it loves water. The tails, on the other hand, they're non-polar and they hide from water. Okay, they do not like water. And then there's also protein still. We won't get to carbohydrates. Now, cell membranes, guys, do have pores. They have holes in it. And this means that they're selectively permeable. Now, the term selectively means that it allows some things and it does not allow some things. And it keeps the other molecules out. And the structure helps it be selective. Guys, why do you think it's important that our membranes are selectively permeable? Answer below. If you said something like, we have to make sure we keep the good things in and we keep the bad things out, you're pretty right. Okay. Now, is there more to it? Can we get more complex? Yes. But if our cell membrane just let everything into it all the time, we continuously be sick, probably dehydrated. We have a lot of issues. But our body is selectively permeable. Our cells, our cell membranes, they're selectively permeable. So they only allow certain things in, they only allow certain things out. Now, does this always work perfectly like this? No. Hence why we still get sick today. Okay. But a lot of the times it does work. And a lot of the times this does work efficiently enough that it keeps us healthy. Okay. It helps us maintain that homeostasis. It keeps us that homeostatic balance. Now, with that being said, when we have different concentrations, things might actually change. We might be able to lose water and gain water depending on what's happening. And we're going to kind of get into that when we get to transports. So wait for that, okay? Now, just as an overview, so we can all see this, we have transport proteins. We have phosphate heads. We have lipid tails. This whole thing is a cell membrane. And remember, it's a lipid bilayer. Lipid meaning it's made out of lipids. Bilayer meaning it's made out of 2 layers, okay? Now, this is a fluid mosaic model. Since these heads are pulling up, these tails are pulling down, these tails are pulling up, and these heads are pulling down, they're repelling each other, okay? So the head's pulling up, the tails are pulling down, so they can't really move anywhere, but they can move to side, okay? They can move side to side really well, and they can pass each other, but they're not rigid in this force that they're going to be stuck in one place, okay? This allows us to have that fluid mosaic piece and allows us to actually bend rather than being a rigid marker, okay? There it is for you. That's just the basics, guys. That is just a little bit of intro into the structure of the cell membrane. Now, we're going to go back to our lab, and we're going to see what actually happened. So give me 1 sec. Welcome back, everyone. Okay, we're going to finish up this lab quick. I've been letting it sit for about 15, 20 minutes now. So I'm taking my carrot out of the fresh water. I'm going to throw it on the scale quick. Carrot now looks like this. Okay, so the freshwater carrot that originally weighed 5 grams, sitting on the scale, and it now weighs 6.5 grams. So finishing weight, I'm going to just write down so I don't forget, 6.5. Now, I'm going to take the carrot out of the saltwater solution, okay, and it now looks like this, okay, and I'm going to weigh this one as well. This one originally weighed 8 grams. This one now weighs 7 grams flat. Now, with your knowledge about cellular membranes and selective permeable, why do you think that these carrots change masses? Okay, I didn't change them. All I did was soak them in water. Okay, I soaked one in fresh water, soaked one in salt water. Why did one gain mass and the other one lose mass? Okay, so just if you guys need a reminder, here's the statistics. Freshwater went from 5 grams to 6.5. The saltwater one went from 8 grams to 7 grams. Okay, so this one lost mass. This one gained mass. Okay, tell me why. Other than that, guys, have a great weekend. Or not great weekend. I'm recording this on a Friday. I apologize. Other than that, have a great Monday. If you guys have questions, please reach out to your teachers, guys. Remember, we're still doing our best every single day. If you guys have anything, please let me know. Other than that, talk to you guys later. Bye.